Hello, everyone, and welcome to this webinar about building better HIV testing, the raw material journey from research to results. Bit of today's agenda. So my name is Teppo Heimo. I will provide you the company Hytest introduction and act as a moderator in this webinar. Our first topic will be about Hytest strategies used for HIV raw material development, held by Dr. Tam. After that, we will hear immunoassay evaluation, Hytest HIV P24 maps and recombinant antigens, which is presented by Dr. He. And after these two topics, we will have a Q&A session about the pre-submitted questions where our presenters are answering those questions. Then a bit of the house rules. Please submit your questions using the Q&A function of this webinar. Also, a recording of this webinar will be available a short while after the webinar is held. And unfortunately, any live questions will not be taken during this webinar, but those submitted to the Q&A will be answered on a handout that is provided you afterwards. So briefly about Hytest, who we are. We have more than 30 years of dedication to providing top-tier immunological reagents for the IVD industry and also for the research community. We are headquartered in Finland and have operations in 3 continents with more than around 150 employees globally. Our products have a solid scientific background, and they have been used in more than 55 scientific publications that we have published. We have an extensive product portfolio featuring selected products as a standard reference material. In addition, we do have active participation in both IFCC and ADLM standardization committee work. We do work closely with various key opinion leaders in the industry, and we have market presence in more than 55 countries. All operations are also compliant with the ISO 9001. What we do is we develop and manufacture world-class antibodies and antigens for the IVD industry, leading to better diagnostics. How we do this is that we first have feasibility, both practical and scientific, for R&D. After that, we proceed to production. We do extensive QC inspection for our ready products. We focus on order handling and then packing and shipment and after-sales support as well. Our comprehensive product portfolio consists of monoclonal antibodies as well as polyclonal antibodies and antigens, serum and plasma. Today, we focus on infectious disease clinical category and mainly focusing on HIV. For HIV, we have developed monoclonal antibodies to be used in antigen tests, as well as several recombinant antigens to be used in serology tests. Some of them are already available, and some will be available quite soon. When you're interested about evaluations, please don't hesitate to contact us through our sales. We are happy to assist you in your product evaluations. Then few words about today's speakers, which is Dr. Natalia Tam and Dr. He. Dr. Tam holds master's and PhD degrees from Moscow State University. She has co-authored numerous scientific publications and patents She has joined Hytest in 2003 and has been involved in different R&D projects. She has expertise in a broad range of disease areas, including cardiovascular and infectious disease biomarkers, as well as many others. She has also deep knowledge in BMP, pro-BMP, and NT-proBNP biomarkers. In addition to these, she has ongoing research focused on HIV biomarkers. She is a leading researcher and project manager at Hytest. Dr. He holds a postdoctorate fellow from University of South California, USA, also doctorate of Medical School of Fudan University, formerly known as Shanghai Medical University. He is an associate editor of the biblical book Immunoassay Handbook, as well as editor of Immunoassay Principle and Application. He is also editor of Chinese-English bilingual immunoassay dictionary. Dr. He has been working and researching on assay principles, technologies, and methodologies and clinical applications of in vitro diagnostics, especially immunodiagnostics, both home and abroad. He has broad achievements in immunoassay development, including biological raw material and reagent design, kinetics and interference studies, as well as assay and process optimization, clinical application study, and troubleshooting. Dr. He is a chief scientist of IVD business unit and director of IVD raw material business in Mindray. So without further ado, give the floor to Dr. Tam, who will present the Hytest strategies used for HIV raw material development. Dr. Tam, go ahead.
Thank you, Teppo, for introduction. Dear guests, good evening. Today I would like to talk about Hytest journey in the development of HIV-specific reagents. In the first part of our webinar, we'll remember the virus structure, its diversity and distribution, and then we'll talk about the ways of P24-specific antibodies and HIV-specific recombinant proteins development for accurate HIV diagnostics. So, HIV, or next, please. HIV, or a human immunodeficiency virus, is a lentivirus which has a single-stranded RNA genome of around 9 kilobases in length that includes 3 major structural genes, GAG, POL, and GAG is first transcribed into unspliced mRNA and then cleaved into transcripts encoding 3 viral core proteins. Matrix proteins, which are necessary for virion assembly and infection, capsid proteins, which form the hydrophobic core of virion, and nuclear capsid proteins, which protect the viral genome by coating and associating tightly with viral RNA, in virions. The POL gene encodes enzymes essential for viral replication, and the N gene encodes the viral surface glycoprotein, GP160, which is cleaved into the surface protein, GP120, and transmembrane protein, GP41, during the process of viral maturation. These surface proteins are essential for virus entry into the host cell. In addition to the major genes, the viral genome also contains regulatory and accessory genes. So HIV is a virus causing AIDS, or acquired immunodeficiency syndrome. Globally, around 39 million of people lived with HIV in 2023. It's important to note here that the aim of the IVD in the field of HIV is HIV detection and as early as possible, not the diagnosis of AIDS. So the accurate diagnosis of HIV depends on the efficiency of detecting the diversity in HIV strains. As much as possible, different strains should be determined with the help of last generation's HIV immunoassays. Where does diversity come from? There are two HIV types, HIV-1 and HIV-2. HIV-1 is divided into 4 groups, where group M is the predominant one, and it's the cause of the global pandemic. Another equally important group is O group of HIV-1, although it only accounts for 1 to 2% of the total HIV-1. Its importance arose when it was discovered outside the Cameroon, especially in Europe and in the USA. All group parents are highly divergent from M group in GAC and N regions and even in POL. This diversity has hindered the diagnosis, monitoring, and treatment of group O infected patients due to the presence of the mutation in a reverse transcriptase enzyme in group O. More than 60% of individuals that live with this virus are faced with the challenge of drug resistance to some antiretroviral therapies. The occurrence of group N and P is very low and amounts to less than 0.1%. HIV also shows marked genetic differences from HIV group M, including over 50% sequence divergence in the genes encoding the envelope proteins. Specific proteins used for HIV assays development starting from the 4th generation are marked on the slide. Such assays can detect capsid P24 protein with the help of P24 specific antibodies and HIV-1 or HIV-2 specific antibodies with the help of recombinant proteins. Usually these are proteins encoded by N gene, and they are GP120 and GP41 and additional synthetic peptides. Speaking about the requirements for modern HIV immunoassays, they need to be designed carefully due to virus genetic diversity, and the main requirement is that these assays should detect both HIV-1 M group subtypes, different serifs, O group strains, and HIV-2 with the help of above mentioned proteins. So talking about P24 specific monoclonal antibodies, let's discard first what is P24 itself. It's a structural protein forming the virus capsid. Of the possible modifications, this protein is presumably phosphorylated. It's conserved between types and subtypes. P24 of HIV-1 M group and O group and HIV-2 are related but not identical. Genetic variation between groups up to 20% and between types from 20 to 35%. This is illustrated on the slide. Existing genetic variations between types and groups confirmed the need to obtain several different P24 proteins for the subsequent development of P24 specific monoclonal antibodies. Serological confirmation of HIV-1, including group M and O and HIV-2 infection, requires broadly specific antibodies. To develop such antibodies, Hytest specialists express 3 different versions of P24 proteins corresponding to group M, O, and P26 from HIV-2 using prokaryotic expression platform. Highly purified proteins were used for animals immunization and further antibody selection. The immunization strategy included P24 antigens from HIV-1 group M, group O, and P26 from HIV-2 to drive the immune response towards recognition of common epitopes within the P24 antigens of both groups of HIV-1 and also HIV-2. All obtained antibodies were tested in pairs in clear sandwich immunoassays, and best combinations met the following criteria. Selected antibodies recognized 3 p24 variants equally. You can see the curves for the representative antibody pair on the graph. This means that selected antibodies recognize shared epitopes or conservative fragments of non-identical p24 proteins. All selected antibody combinations recognize the following HIV virus subtypes included in the first WHO International Reference Panel for HIV 1 p24 antigen. And they also recognized first WHO International Reference Reagent for HIV-2 P26 reagent antigen with a high efficiency. Based on additional research, several MAPS combinations can be suggested for the development of HIV P24 immunoassays. MAPS could be used on ELISA and CLIA platforms. HIV infection, HIV-1 or HIV-2 can be detected, including group O, different serifs and other subtypes, using only one antibody pair or 2 monoclonal antibodies instead of a set of monoclonal antibodies, 3, 4, or a special set used for HIV diagnostics by several IVD companies. So you can find 3 different P24 specific antibodies in Hytest catalog. Our next part is devoted to the development of HIV-specific recombinant proteins for HIV antibody detection. The definition and selection of proteins for HIV antibody detection are generally based on high immunogenicity of the protein or epitope, early rising of specific antibodies. It's important when the protein represents diversity, and the protein sequence should include key immunodominant regions. So these are the general recommendations for protein selection. The proteins that meet the above criteria are those encoded by ENF gene. These proteins represent major molecular targets of HIV-specific antibodies detected with HIV immunoassays. Epitopes for human antibodies isolated from infected individuals have been characterized very well. Found antibodies and their specificity are shown on this picture. The fragments under which the brighter squares are located indicate the most frequent occurrence of specific antibodies. So let's have a look at these major proteins. The first one, the P120. On the right side of the slide, the organization of the virus spike formed by GP120 and GP41 is presented. GP120 is an envelope glycoprotein. Exists as a trimer, which is non-covalently interacted with other envelope protein GP41. Sorry, it has several constant and variable regions. V3 and V4 regions interact with CD4 to trigger viral entry. High diversity between GP120 from HIV-1 and 120 from HIV-2 was documented. The envelope glycoprotein is heavily glycosylated with around 30 N-linked glycosylation sites. Glycan represent up to half of the protein mass and occupies from 50 to 70% of the surface. And DARS has a major effect on the antigenicity of the protein. The dense glycan shield contributes greatly to the heterogeneity of the recognized structures on the virion surface. During chronic infection, the nth protein can lose or gain and linked glycan sites, and it's thought to be a special immune escape mechanism of the virus. So, next protein is GP41. It's a transmembrane glycoprotein with several functional regions. In mature form, it's a trimer. It hasn't so many sites of glycosylation as GP120. Only 4 sites of N glycosylation were determined. According to the previous slides, GP41 has several immunodominant regions along its ectodomain and practically no regions for producing antibodies along its transmembrane and C-terminal fragments. As we can see on the right side, next please, there is high diversity between GP41 of HIV-1 and its analog GP36 from HIV-2. According to the available info, presumed design of HIV-specific raw materials from 2 global IVD companies are as follows. Different proteins such as GP41 from N group, combination of GP120 and GP41, GP36 in recombinant forms expressed in E. coli cells, and also HIV-1 or group-specific synthetic peptide derived from immunodominant region. Design of Hytest raw material was also based on available data. Genetic diversity was taken into account, consensus sequences were used, and also additional approach were used when the same protein used for coating and detection and was obtained by different expression platforms. So the maximal diversity in one recombinant protein was achieved by using consensus sequences of different fragments of GP41 and GP120 from HIV group M and group O fused together. The scheme of this construct is presented on the slide. Such protein contains the largest amount of immunodominant regions. The approach where the same protein used for a double antigen sandwich produced in 2 different expression systems, E. coli and mammalian cells, should have led to reduced risk of interference when used in the assay. This idea is illustrated graphically below on this slide. Briefly, if antibodies specific to some cell host proteins are presented in the sample, no false positive signals will occur since the detection antigen will have different possible composition of impurities. The features of such HIV-specific proteins, fused proteins, are presented on this slide. In case of prokaryotic expression, recombinant fused protein was expressed with a protein tag with no post-translational modifications. The protein was partly refolded and also has reactivity with HIV-1 MNO group blood samples according to the HIV test manufacturers, and protein shows acceptable stability under different temperature conditions. As for the protein expressed in mammalian cells, it was also expressed with protein tag partner. Its post-translational modifications like glycosylation could be similar to HIV protein circulating in the blood of infected people. It's a soluble protein. This protein has reactivity with HIV-1 M and O group blood samples according to the HIV test manufacturers, and protein shows high stability under different temperature conditions. For other HIV proteins expression, the same approaches were used as it was discussed above. Mammalian expression platform was used for the following proteins expression for GP41, GP36, and GP120 fused protein combined with M and O group fragments. All these proteins were expressed with protein tag partners. Post-translational modifications like glycosylation of these proteins could be similar to HIV proteins circulating in the blood of infected people. All proteins are soluble. All of them have reactivity with blood samples of corresponding group, according to the HIV test manufacturers. And all these proteins showed high stability. So HIV-specific proteins that were discussed are presented in this table. All of them could be used for HIV antibody assays or combo assays development in various combinations. Thank you for your attention.
Thank you, Dr. Tam, for the very interesting presentation. Next, we will have a presentation from Dr. He about immunoassay evolution, Hytest HIV, P24 monoclonal antibodies, and recombinant antigens. Dr. He, please, floor is yours.
Thank you, Director Eckelham. Hello, everyone. Following Dr. Tam's presentation on Hytest's new HIV monoclonal antibodies and recombinant viral proteins, I'd like to show you the preliminary performance results using the new HIV maps and the recombinant viral proteins evaluated as a prototype assay on mandatory automatic chemiluminescent immunoassay system. As all you know, HIV immunoassay has transformed the screening of the viral infection and its diagnostics by innovating viral antigens and assay architectures. For IVD companies like us, this evolution reflects critical advances in assay sensitivity and specificity. Next slide, please. I will first present a snapshot of evolution of HIV immunodetection technologies in the past 40-some years, then show you our evaluation results for P24 antibody pairs and the recombinant antigens combinations using the automatic chemiluminescent immunoassay system. First, see here the HIV immunoassay evolution. Basically, the history of HIV immunoassay is the history of sensitivity evolution and their promotion. You know, after infection of HIV, the viral RNA can be firstly detected at the average time of 11 days, and followed by the appearance of P24 antigen and anti-HIV IgM antibodies, which can be detected at the average time of about 17 to 22 days, respectively. The first generation you see here, the first generation assay was actually using the viral cell culture lysis, the total cell lysis, as antigen to capture HIV-specific antibodies using indirect format for HIV IgG detection. The negative window time actually is between the viral infection and the detected time is about 8 to 10 weeks, so about 3 months. The crude viral lysis used in the first generation assays contain, the specific antigen like GP120 and P24, but also host cell debris, cellular proteins as well. The non-specific cellular proteins have caused unacceptably high false positive results. That's the problem. So the second generation assay switched to the recombinant viral proteins, which can detect both HIV-1 and HIV-2 IgGs. The window is reduced then to 4 to 6 weeks. The purified antigens have largely solved the specificity issues for the first generation assay. And then in early 1990s, the third generation came up using recombinant protein and also the synthetic peptides for detection of both IgG and IgM, and also group O. The window is therefore reduced to 2 to 3 weeks. The third generation was using the dual antigens and sandwich format, like what just Dr. Tan introduced, enabling IgM detection besides IgG detection. This cut the serial conversion window to like 22 days. So in the late 1990s, the P24 antibody pairs are added to the third generation assay to detect the P24 antigen. Besides detection of the viral IgG, IgM antibodies, this is becoming the fourth generation assay. The window is further cut to about 2 weeks. Now there is a so-called fifth generation. On the basis of fourth generation, separate reactions in different vessels reporting the results for HIV-1, HIV-2 antibodies, and also P24 separately. The fifth generation's kind of multiplex assay can really answer the questions such as, is it an antigen or antibody, acute or chronic? And by physically separating the signals, the laboratory stage-specific diagnosis without the reflex tests, which is the additional advantage for the first fifth generation. Next slide, please. Antibody is the key raw materials in immunoassay. It's about like 50% of IVD product records actually can be traced back to raw materials directly or indirectly. And raw materials, especially monoclonal antibodies, are difficult to replace and can be very costly to change the maps in registered IVD products. And also, antibody in good quality has to have at least good purity, stability, plus defined epitope information, affinity, high affinity. There are not so many good antibodies in the market, unfortunately. And next slide, please. That's why, we have tested, Hytest newly designed and developed maps in order to improve the current P24 HIV P24 detection. I'm sharing you our very preliminary results here. Next slide. First, the assay design. This assay, prototype assay design, basically using the sandwich format to evaluate the monoclonal antibodies. It's based on the current the new system here, the pan-immunizant immunoassay system. In the first step, the P24 specific maps are coated onto the paramagnetic particles. These particles capture the corresponding HIV-specific antigens, that's P24, from the sample. The following a washing step, the captured HIV antigen are detected in the second step using the alkaline phosphatase conjugated monoclonal antibodies. The LP actually catalyzes the substrates within the immunocomplex bound to the particles, generating a signal that is measured through a chemiluminescent reaction. For antigen assay, I will show you later. Basically, we are using the same two-side sandwich format, but just using recombinant proteins replace the monoclonal antibodies. Next slide, please. So the first results are this signal to noise ratio. This is important to us. Signal to noise ratio is a very quick check for testing of testing analytical sensitivity. The higher the ratio, likely the higher the analytical sensitivity. So in this study, totally we choose 23 monoclonal antibody pairs for the samples, including Hytest HIV1 M and O group, P24 antigens, and also HIV2 P26 antigen, and the P24 WHO standard. You can see here the signal to noise ratios are high, indicating that all the pairs can detect each of the antigens at the concentrations of HIV1 at 10 mg per ml, HIV2 20 mg per ml, and the P24 WHO standard is at 2 IU per ml, which is consistent with Dr. Tan's results just showed. Next slide, please. Okay, then for the limited detection of the study, that's basically the analytical sensitivity as well. And 17 maps out of the tested 23 pairs were tested. Results showed you the LOD of WHO standard are from the 0.5 to 0.65 IU per ml, which is very sensitive. Meets the requirement of EU Commission implementing regulation, which is less than or equal to 2 IU per ml. Most of the maps showed a greater detection capability for HIV P24 and also P26 antigens than the results generated on the groups in comparison. Please pay attention here. The HIV HIV2 here, there's P26. There's one group actually missed the P26 here in the comparison. Okay, next slide, please. To come to the specificity, we then evaluated 7 map pairs using 1,000 to more than 6,000 samples. All map pairs showed that the specificity more than 99.5%, meeting the requirement of EU Commission implementing regulation as well. And next slide. Now let's take a look at the results for the reference panels. That's first panel. We select 2 pairs of the Hytest pairs for the evaluation. Both pairs showed higher COI, the cutoff index, for all the P24 WHO genotype panel, 210, and also the Hytest P24M and O groups at 2 pg per ml, and the P26 at 20 and 10 pg per ml. Okay, and then next slide. Okay, we also tested the 2 pairs for the P24, P26 reference panel, and from a third party, with the common, circulate recombinant force and the other groups, very representative CRFs here. And in general, most of the map pairs showed higher sensitivity than the compared groups in terms of COI values. Okay, next slide. Now I move to HIV recombinant protein evaluation results. Next. The first is a sensitivity study listed on the right upper corner of the slide are the recombinant antigens we have evaluated. This data showed the recognition of HIV-1 using the 2 pairs of antigens on our system, and the 3 pairs of antigens recognizes HIV-2 antibody positive samples compared with 1 or 2 IVD combo assays. Next slide. Further, we evaluated the specificity of the 2 pairs of recombinant antigens of HIV-1 antibody positive samples for HIV-1 positive samples. The total sample number is 2,957 for the first pair. Results showed 99.86% for specificity, while the total number of the sample of 5,901 is tested for the second pair antigens, showed 99.83% specificity. This is for HIV-1. Next slide, please. And then for HIV-2 antigens, we evaluated the specificity of the 4 pairs of recombinant antigens for HIV-2 antibody positive samples from 750 to 6,683 of total numbers. And results showed 99.87% to 100% specificity. Next slide, please. Now, let's see the results of HIV antibody antigen combo evaluation. Basically, this is not a validated prototype. Okay, so it's kind of a combo here. We designed 2 HIV combo prototypes using different antibody antigen pairs from Hytest, and both can detect all subtypes samples tested here, you can see. And one of the IVD combo compared here missed 1 samples as highlighted in red here. The Hytest HIV antibody antigen combo use here, actually, we can see are able to detect all different genotypes, quite comparable to the groups in comparison, if not better. Next slide, please. Look at the HIV antibody antigen combo evaluation results for the serial conversion panel, which to me is a very critical performance in terms of assay clinical sensitivity and also clinical value. For total 22 serial conversion panels, it's with 222 samples, total samples. The HIV combo 1 using the Hytest raw materials again showed 97 reactive samples, and the HIV combo 2 demonstrated 100 reactive samples, while the control groups showed 96 reactive. Therefore, Hytest HIV combo assays demonstrated comparable, very comparable sensitivity results as compared with the global companies, one of the global companies here, with the serial conversion panel. Next slide, please. Lastly, let's take a look at HIV combo evaluation results for specificity. For a total of 4,272 clinical samples with a variety of disease statuses. See the samples listed on your right, including the interference samples. Combo 1 showed a specificity that's highlighted right here. You can see 99.93%, and the combo 2 showed 99.86%. 0.91% respectively. Therefore, both HIV and AG combo prototype assays using Hytest raw materials demonstrated equally high sensitivity and specificity. Next slide, please. So in summary, the evaluated HIV P24 monoclonal antibodies using mandatory clear system exhibit high sensitivity and specificity in the detection for both P24 and P26 antigens. This is particularly demonstrated on the 22 serial conversion panels. And also the assessed Hytest recombinant viral proteins using the clear system demonstrated high sensitivity and also specificity with the clinical samples as well. Next. And with that, thank you for your patience.
Thank you, Dr. He, for very intriguing presentation. Next, we will move to the Q&A session with the predetermined questions that we have received during the registration. First question goes, can you explain your quality control process that you are using for your Hytest products? And I think this question goes for Dr. Tam. Please go ahead.
Thank you. Thank you for the question. I guess I can answer as follows. Hytest quality control process can be described as like this. We use raw materials, so chemical reagents, only from suppliers who are approved on our list of acceptable suppliers based on quality, reliability, etc. We follow performance of our suppliers continuously. Incoming inspection is performed on their materials. This is in many cases a document review to confirm acceptable specifications, for example. Only raw material from product-specific list of reagents can be used in manufacturing and quality control. The reagents on the list are carefully selected and qualified by our R&D before product launch. All product lots are QC tested to confirm that lots correspond to internal specifications and pass predetermined QC acceptance criteria. Hytest uses a broad variety of quality control tests for produced proteins, such as appearance control, purity determined by means of SDS page or capillary electrophoresis, and also proteins immunoreactivity determined in sandwich immunoassays, which use protein-specific polyclonal or monoclonal antibodies. Thank you.
Thank you, Dr. Tam. Dr. He, do you have any additions you would like to share?
No, not to this point.
Okay, thank you. Let's move forward to the next questions. Why develop fifth generation assays when the fourth generation assay is good enough? I think Dr. He would be the best to answer to this question. Please go ahead.
Thank you for the question. Fourth generation definitely is good enough, but it only tells you the IgG or antibody positive is very qualitative. It doesn't stage infection. The fifth generation really can show some additional benefit. first, it can cut the confirmatory testing costs by 40%, and can really flag acute cases for urgent antiviral therapy. And in the market, like in Europe and the United States, where the molecular diagnostics testing are very expensive, this is a key value proposition, I think.
Thank you. It very much seems so that there are kind of pros to develop fifth-gen assays as well. Next question. How critical is recombinant viral protein stability in fourth generation or fifth generation regions? And I think this goes to Dr. Tam, who will be able to provide answers for the region part, at least.
Yeah, thank you. I can start from the view of R&D. It seems that stability of HIV-specific proteins is a critical point in HIV-specific reagents. Using mammalian expression platform helped to obtain proteins that are quite stable during production process, during storage, and upon handling. We, from our side, optimize protein production and storage conditions to ensure their maximal stability. Also, I guess post-translational modifications and the presence of protein tags contribute in pretty good stability of HIV proteins expressed in mammalian cell line. So I think that's all from my side.
Thank you. Thank you, Dr. Tam. Dr. Hurd, do you have anything to add?
Yeah, probably I can talk about some points from the assay development perspective. And first, we believe recombinant viral proteins sometimes it's very vulnerable to aggregation and sometimes degradation as well. And besides what Dr. Tan just showed, the Hytest has done to achieve the high stability for raw materials. However, for us in assay design development, because we're really coating the antigens or sometimes for the conjugation as well. So we can modify the antigen chemically. There are something we actually pay attention to the raw materials and also more for the reagent stability. In this way, actually, we kind of focus on the reagent design, the reagent formulation, and we also optimize the process and also sometimes really see the some issues for the or potential issues for the susceptibility of the particular antigen in the presentation of our region. We have to work on the how to enhance the stability using the reagent design and by adding the big molecules and sometimes working on the detergent and stabilizers and also keep looking for that lot. I think that's very important, particularly for the HIV assay, which requires high sensitivity for antigen test.
Thank you. Thank you. I think that's very, very thorough answer on this question. let's move forward. The next question is, can synthetic peptides replace recombinant antigens? And I think this one should go to Dr. Tam. Please go ahead.
Thank you again, for this question. From my point of view, synthetic peptides can't fully replace recombinant proteins. As a rule, HIV proteins have several epitopes or antigenic determinants for antibody production on their surface. Epitopes can be either continuous or linear, in which several amino acids usually from 6 to 8 amino acids are present in the sequence, or discontinuous, in which the amino acids that form the epitope are brought together by the three-dimensional folding of the protein. Even though an epitope constitutes only a relatively few amino acids, its reactivity with an antibody is influenced by amino acids not included in their epitope or by the presence of post-translational modifications, which may also be located outside the epitope. Moreover, it's easier to take into account the genetic diversity while using a recombinant protein, not the single peptide. More epitopes you have in your protein sequence, then more likely you will be able to determine the presence of specific antibodies. I believe that in some cases, to ensure high sensitivity in the context of recognizing a wider variety of strains, a specific peptide can be added to the test system, but it's not possible to replace the test system with peptides completely. But as we know, there are approaches in IVD which combine the recombinant proteins with the GP41 specific peptides also. Thank you.
Thank you. Thank you very much. Next question. Is it necessary, and if so, how often and in what way should IVD companies or test system manufacturers evaluate or verify the current HIV assay for its ability to detect newly emerging HIV strains? And I think this is a perfect question for Dr. He, so please go ahead.
Yeah, thank you. I really think this is a very good question, and very critical and it's significant to me. HIV, is a highly genetically variable virus, and circulating recombinant forms continue to emerge and spread. The IVD manufacturers, I think, should evaluate their HIV immunoassay reagents for the ability to detect kind of emerging HIV new strains and mutants, and also new CRFs on a regular RESC-based cycle. However, we don't know, at least I'm not sure if there is a fixed interval requirement. But I think I would suggest the following approach for the evaluation of the new detection for the emerging strains and also the CRFs. First, I think we need to keep abreast of new HIV group M subtypes and new CRFs reported, and any mutations that could affect antigen-antibody binding, updating the RESC analysis based on the findings to ensure the assay vulnerability is identified early. And then I think continuously monitor field complaints, hear from the customer complaints, feedbacks, and the proficiency testing results, and the regulatory alerts, and the scientific articles, If trends suggest reduced sensitivity in certain geographics or certain population or sample types, I think this should trigger immediate reassessment or reevaluation of the assay. And then manufacturers should perform formal revalidation if high-risk variants emerge. This evaluation should use panels that include the recent clinical isolates and the newly identified CRFs to confirm the assay still meets the sensitivity and the specificity claims. And lastly, I think lot-to-lot verification testing with HIV reference panels should continue routinely as required by regulators to ensure production consistency. Any changes in antigens, reagents, or manufacturing process also require targeted reevaluation of the strain. I believe that the necessary revalidation provides a robust framework for maintaining reliable HIV detection across evolving viral strains and the emerging CRFs. Thank you.
Thank you. Thank you very much. I think we are kind of running out of time, so let's wrap up today's webinar. I want to thank our speakers, Dr. Tam and Dr. Herb, both equally for great presentations and to bringing us a lot of information about HIV and the new assays around HIV. If you are interested in evaluating or upgrading HIV assay, we are happy to collaborate with you and help you to develop new assays for HIV detection. Like I said, this webinar will be published afterwards as a recording, and handouts will be provided for all participants. With that, I want to thank all for the attention and wish everyone a good day. Thank you for participating.
Thank you.
Thank you.